Using this protocol, cell migration and invasion can be unveiled under real-time conditions, enabling cell kinetics under loss or gain of gene function and drugs to be determined. Unlike other methods, no staining, mechanical cell damage, or fluorescent markers needed. Most importantly, the real-time cell kinetics can be determined in an effective manner.
When the U118mg cell line culture reaches 70 to 80% confluency, wash the cells with PBS before treating the cells with 2 milliliters of 0.05% trypsin EDTA. After 30 seconds at 37 degrees Celsius, stop the reaction with 10 milliliters of fresh culture medium and transfer the detached cells to a 15 milliliter conical tube. After counting, collect the cells by centrifugation and resuspend the pellet at a 6 times 10 to the 5th cells per condition concentration in the appropriate volume of fresh medium. Transfer 6 times 10 to the 5th cells into 1 microcentrifuge tube per condition and add 800 microliters of Delbeco's PBS to each tube. After centrifugation, resuspend each pellet in 60 microliters of resuspension buffer R and 6 micromolar of the siRNA of interest. Mix each tube with gentle tapping before electroporating 10 microliter aliquots of each cell suspension with 3 10 millisecond 1350 volt pulses. After each treatment, pool the electroporated cells from each condition in individual 5 milliliter aliquots of fresh culture medium.